Thank you. I'm delighted to introduce the next speaker, one of our T32 Psychology and Medicine postdoctoral scholars, Dr. Lauren Whitehurst. Dr. Whitehurst obtained her bachelor's and master's degree from James Madison University and her PhD in cognitive psychology from UC Riverside. Her research examines how sleep processes influence short-term and long-term effects on cognition and takes seriously the idea of how the underlying neurobiological milieu during sleep has direct pathways to cognitive outcomes. When Dr. Whitehurst first applied last year to our T32 training program, we were all impressed with her application and her publications, and similarly, when we interviewed her, we thought she'd be a great addition to our group. But it turns out that we dramatically underestimated how great she is. It turns out in the short 6 months that she's been here, here's an incomplete list of her accomplishments. She's completed a week-long daily diary study with more than 100 participants, examining how sleep and daily blood pressure influence memory. She launched a study in the sleep lab with Andy Crystal and Eric Prather, examining how exposure to racial discrimination affects neural and autonomic responses during sleep. She submitted a RAP grant. She's about to submit a manuscript on how sleep influences cognitive processing in older adults. And she designed a complete cognitive task module that's been integrated in an app-based study called My BP Lab that measures stress and blood pressure on smartphones. She is a whirlwind of activity, but always thoughtful and precise in her work. We feel so lucky to have Lauren as part of our group in health psychology and are delighted that today you get to meet her and hear about her work as well. My pleasure to welcome Dr. Whitehurst, who will be presenting on the impact of psychostimulants on sleep-dependent memory in healthy adults.
So we're going to hear a lot more about drugs to start. And thank you so much for that warm introduction, Wendy. I really appreciate those kind words. And thank you all for being here. It filled up. My back was turned. I didn't realize how many people came in. And so again, the title of my talk, I changed it just slightly to the impact of psychostimulants on sleep and cognition in healthy adults. And when I say psychostimulants, I'm really referring to prescription stimulants, amphetamine-based stimulants, that are usually prescribed for things like attention deficit hyperactivity disorder, but over the last 2 decades have been increasingly being used off-label by healthy individuals under the guise that these drugs are actually going to enhance their cognitive ability. And this type of use is not uncommon. In the last 2 decades, it's been about a 10-fold increase in the use of these substances for this purpose. Conservative estimates here in the U.S. suggest maybe upwards of 11 million people are using these drugs for this purpose. And up to about 35% of college students endorse using psychostimulants off-label for cognitive enhancement. And this use isn't completely misguided. These types of psychostimulants they impact neuromodulators that are pretty well implicated in learning and memory. They upregulate norepinephrine and dopamine directly at the synapse, and they also kind of indirectly impact acetylcholine, a neurotransmitter highly implicated in memory processing. And additionally, these drugs have been shown to help to boost cognition, specifically in military contexts where individuals are up for 24 hours or plus, and in shift work contexts when individuals are awake when they'd rather be sleeping. These drugs have been shown to help individuals maintain alertness, promote sustained visual attention, spatial planning, and spatial working memory. However, in more healthy contexts, these drugs actually have a little bit more mixed results. So they've been shown to be differential across cognitive domains. So if you're looking at attention or the allocation of your processing resources to external stimuli, you see very different effects than if you're looking at working memory or declarative memory. And there's also been studies suggest that there's differences within cognitive domain, where some studies suggest that these drugs actually might help declarative memory. Other studies suggest that they have no effect on declarative memory, and still others saying that they might actually impair declarative memory. So these differential effects both within and across cognitive domain led me to think that there might be something else that's influenced by, that influences cognition, but is also influenced by these cognitive stimulants. And that led me directly to sleep. So we know that one of the main reasons individuals are using these stimulants is to prolong their wakefulness, is to keep them, to stave off sleep, keep them awake and productive and on task. And individuals with a history of this off-label psychostimulant use report decreased objective sleep quality, increased reports of nighttime sleep disturbance, and indeed these psychostimulant drugs are there to keep you awake longer. We know that if you give them to people before sleep, they have reduced quality and quantity of nighttime sleep. This is measured with electroencephalographic activity or polysomnography, the kind of gold standard in sleep science. However, sleep is really important for cognition. There's been a wide-ranging literature suggesting this. Suggesting that sleep losses lead to reduced daytime vigilance, attention, and executive function. And different stages of sleep actually have differential outcomes for learning and memory outcomes. Specifically, slow-wave sleep being shown to be really good for learning stabilization. So as you walk throughout your world and you bring in all this information and you attend to all these stimuli, you go to sleep at night and then you can stabilize that information. Make it really nice and set so that later when you need to come back and get that information, you have it there to retrieve it. And indeed, time spent in slow-wave sleep is related to better memory after sleep. Slow-wave sleep have also recently been shown to support working memory training. So being in these tasks, in these protocols where individuals are training on these different types of working memory paradigms, kind of like that brain training literature that we're seeing growing, slow-wave sleep actually seems to be quite important. That increases in slow-wave sleep are related to better working memory outcomes. And then lastly, rapid eye movement sleep, kind of like the superstar of the sleep literature, has shown that this stage is really good for reanalysis and integration of new information. So taking that information that you learned, this new knowledge information that you learned, and putting it with prior knowledge. And indeed, time in REM sleep is associated with increased insight into problems and better creative and associative memory processing. And so this really leads me to the questions that I have for today. So my first question is, do psychostimulants, these types of amphetamine substances, enhance cognition in healthy adults? I'm going to be exploring that question in two different studies. In the first study, I'm going to be showing you data from sustained attention, working memory, two domains that I would expect to be enhanced by cognition by these stimulants. And then study two, I'm going to be looking at declarative memory. And in this study, I'm going to be kind of probing that sleep question a little bit further. I'm also going to be asking, what is the time course of these effects? What are the immediate versus long-term effects of these drugs on cognition? Do these drugs disrupt sleep? And lastly, if they do disrupt sleep, what are the long-term cognitive costs of that sleep disruption? So in the first study, I'm going to be looking at two different cognitive domains. The first is attention, and the second is working memory. For this study, I used a double-blind, placebo-controlled crossover design. I brought young, healthy subjects into the lab. They had no neurological, psychological, or other chronic illnesses, and they all reported regular sleep patterns before the study, and we actually made sure that they kept those rhythms, those sleep patterns throughout the study with both actigraphy watches and sleep diaries. And all of our subjects in the study were quite drug naive, so they reported less than 2 lifetime uses of any sort of stimulant medications, and none in the last year. The drugs for the study that we used were dextroamphetamine, a 20 milligram dose, pretty typical stimulant used for chronic fatigue and things like that in the literature. And we also use a placebo control, just a non-psychoactive substance. For this study, we brought people into the lab at 8 a.m. We got task baselines for both the attention and the working memory task. After that, we then administered the drug of the day, which was either the stimulant or a placebo. 75 minutes later, which was kind of peak plasma for the substance, we then tested them on that information that they learned at baseline. 12 hours later, we then tested them again to kind of get at that time course. All subjects slept in the lab with polysomnography, so we hooked them up completely to EEG so we can get sleep architecture throughout the night. And then the next morning, we woke them up, and we had them take those two tasks one more time before leaving the lab. And again, just to make sure that we're clear, this is a crossover design, so everybody got both the stimulant condition and the placebo condition. So for attentional processing, we use a multiple object tracking task. In this task, basically, subjects were asked to keep their eyes fixated on the center dot while tracking these flashing targets. So all the targets would move on the screen. At the end of the trial, 2 dots would highlight red, and subjects would be asked, were these the 2 dots that you saw at the end of the trial or not? We had 2 different conditions. The first was a unilateral condition, where our targets were on one side of the fixation. And the second was a bilateral condition. We had targets on both sides of that fixation. Again, targets would flash, everything would move. Subjects' eyes were fixated on that center dot. At the end of the trial, dots would highlight. Were these the same dots that you saw at the beginning of the trial? So slightly more difficult condition than the bilateral condition. I can hear from the kind of grunts in the room. So for this task, we had baseline. So we first brought people in at baseline, and we gathered speed thresholds. So we wanted to know the speed at which participants could perform this task at 80% accuracy. And then for all subsequent tests, we set the task to those speed thresholds and calculated their accuracy at those speeds. And that just allowed us to use this type of task in a repeated measures format. The second task we used in this protocol was a working memory task. For this task, we had subjects fixate on a center cross, again computerized. They then saw an equation, and they were asked to tell me if this equation was accurate or not. And then they saw a letter. And the participant's task was to remember all of the letters that they saw in the exact order that they saw them. And so subjects would continue to see a series of letters and equations, roughly a string of 4 to 8 letters, before they would then be prompted to respond with all the letters that they saw in the exact order. For this task, we separated our conditions and kind of blocked them into 4 to 5 letters, which were our easy trials. Subjects did quite well in those. And then 6 to 8 letters, which were our hard trials. Subjects had a little bit more variance at the performance level. For this task, we looked at percent correct, which is just accuracy of the amount of letters that you could get, and then also percent change from baseline. So let's get into the results. So first, for the attention task, I'm going to first show you data from that 75-minute condition. Just to kind of orient you to these graphs, I'm showing you violin plots. And how you can interpret these is that this little center diamond, which doesn't look much like a diamond right now, is the median. And then these are just regular box and whisker plots with this black rectangle being the interquartile range. And then the shaded region, which is white for the placebo condition and gray for the dextroamphetamine condition, is basically just subject density, the amount of subjects in each of those, at each performance area. And what we're showing you here is that in the acute condition, when the drug is still online, 75 minutes after drug ingestion, we see that stimulants do boost attentional processing. About 4% for both our unilateral condition and our bilateral condition. 12 hours after drug ingestion, we don't see a benefit for the unilateral condition, but we do still see a benefit for our bilateral condition, suggesting that subjects are actually learning something about this task that's then transferable to later tests of attention. For our working memory, we did not see an impact. These bars could not be more similar between our stimulant and our placebo condition. So there was no immediate impact of the stimulant on working memory. At our 12-hour condition, we again did not see any benefit there. We do see a main effect for difficulty, just that our easy trials were easy. And we see this both with percent correct and also percent change. So we don't see any difference between any of these change scores either. We start to see this kind of little benefit here. It's not significant, but I did keep my eye on that for the future and for my next analyses. So how did the stimulant affect sleep? Again, we gave the stimulant at 9 a.m. in the morning, and we're looking at sleep after 11 p.m., and we're trying to see if the stimulant still has an effect. So a full day. And we did find that the stimulant universally disrupted sleep. So looking at our main sleep kind of outcomes, total sleep time, minutes in each sleep stage 1, stage 2, slow wave sleep, and rapid eye movement sleep. Wake after sleep onset, sleep latency, the amount of time it takes you to get to fall asleep once you're in bed, and then sleep efficiency, the amount of time that you spend asleep when you're in the bed. What we see over each of these conditions is that the stimulant reduced that sleep outcome. With the exception of stage 1 sleep, which is kind of indicative of waking up a lot, so we actually want less stage 1 sleep. And then also with the exception of wake after sleep onset, which again, the stimulant increased. So again, just kind of generally impaired our nighttime sleep. So what did that do for our cognition after sleep? For attention, we actually didn't see much of a problem. So the stimulant did not seem to decrease sleep. If anything, in this bilateral condition, we're still seeing a little bit of a trend that the performance is still kind of a little bit better in that stimulant condition compared to placebo. For working memory, here we start to see a little bit of a different picture. So what we see is that in test 3, which is 24 hours after drug ingestion, we see that there is a benefit in the placebo condition, such that people are getting better after sleep compared to test 1, that you don't see in the stimulant condition. Sorry about that. Thank you. Thank you so much. Okay, and then looking at, just to kind of, for time's sake, looking at percent change, this actually becomes a little bit more apparent here. So we're looking at percent change from baseline, and we see that in test 3, after sleep, there's this benefit in the placebo condition that we do not see in the stimulant condition, suggesting that there might be something that sleep is actually helping with this working memory training that goes away after the stimulant. Okay, so now I was really interested in what about a really sleep-friendly process? So looking at that declarative memory task that I mentioned earlier. And so for this study, I had slightly different questions, similar to study 1. But here I was going to be asking, first, do stimulants enhance declarative memory processing? Second, is there a post-sleep cognitive cost to stimulant enhancement? And here I'm really going to be probing, is this cost due to stimulants' impact on sleep? So I'm really going to be trying to figure out if it's a sleep mechanism. And then lastly, can we recover or restore sleep, in this case with another drug, a hypnotic, and then bring back memory? So for this study design, it was also a double-blind, placebo-controlled crossover. We brought young subjects into the lab. Half of them were female. They were all very healthy, similar to study 1, and the same types of exclusionary criteria that we had in study 1. For this study, we had 2 different active substances. The first was 20 milligrams of dextroamphetamine, similar to study 1. And the second was 10 milligrams of zolpidem, which is also known as Ambien. It's a really commonly used hypnotic, and it has previously been shown to enhance sleep, specifically slow-wave sleep, like I mentioned earlier. And again, this was also placebo-controlled. For the task here, we used the declarative memory task, the emotional picture task. We wanted to kind of play with the valence of the memories. There has been some studies to suggest that stimulants might have a specific impact on emotional memory, so we wanted to kind of be able to look at that here. For this task, we used pictures from the International Affective Picture System, which is a widely used database that has kind of normative ratings for both valence and arousal, and we extracted those for this task. At encoding, we had individuals look at 48 images. 20 of those were negative, 28 of those were neutral. And after they see the image, we just asked them to tell us, was this image taken indoors or outdoors? At each subsequent test, individuals saw 40 images total. They saw 20 images, 10 of those that were old, that they had previously seen during encoding. 10 of those were negative and 10 of those were neutral. And they also saw 20 new, completely new images. 10 of those were negative and 10 of those were neutral. And for our outcomes here, we looked at accuracy, just percent correct, forgetting, percent change between test 1 and test 2, and then D prime, which is just a kind of a measure of memory integrity. So the amount of these images that they saw, that they correctly identified seeing, minus the amount that they did not see, but they incorrectly identified seeing. For this protocol, we brought subjects into the lab at 9 a.m. We administered our drug right then. That was either the stimulant pill, dextroamphetamine, or placebo. 75 minutes later, we then had them encode the drug. So they're doing their encoding while they are on the drug substance. Sorry, we had them encode the task. They're doing their encoding while they're on the drug substance. 12 hours later, they had the first test of that material. Shortly after, we then administered the second pill of the day, and that pill was either the hypnotic, zolpidem, or placebo. All of them slept overnight in the lab with electroencephalography so we can get sleep architecture. Next morning, we woke them up and gave them the second test of the second test. Importantly, we had 4 different drug conditions here. The first was placebo-placebo, where they got placebo in the morning and then also placebo at night before sleep. The second is dextroamphetamine placebo, where they got the stimulant in the morning and the placebo at night before sleep. The third is dextroamphetamine zolpidem, where they got the stimulant in the morning and then the hypnotic at night before sleep. And then lastly, the placebo-zolpidem condition, where they got the placebo in the morning and then the hypnotic at night before sleep. We allowed 1 week between each of these experimental visits just to allow for drug washout, and all subjects went through all 4 of these drug conditions. So first we're looking at the pre-sleep memory, so the memory before they went to sleep. And what we're seeing here is a very traditional emotional memory benefit. So what you see is that individuals remembered the negative images slightly better than the neutral images. That's pretty typical, shown in the literature quite often. However, when we look at our stimulant condition, we see that emotional memory benefit go away. And we see this increase in neutral memories, the same salience of emotional memories, suggesting that the stimulant's actually boosting these neutral memories to that same level. What do we see at sleep? The same thing. The stimulant still disrupted sleep. Again, we're giving these drugs at 9 a.m. in the morning, and we're still seeing these kind of broad sleep disruptions. So again, after the stimulant, we saw increased stage 1 in wake after sleep onset, decreased sleep efficiencies, total sleep times, and slow wave sleep and REM sleep. Did this affect post-sleep memory? It actually did. So what we found here is that 6% increase in neutral memories that we saw in encoding was actually the same neutral memories were actually hurt here in that stimulant condition. So we saw a 12% decrease in memory here compared to the placebo condition. Was this related to sleep outcomes? It indeed was. So first we see that the amount of time it took people to get to sleep was related to this memory outcome. So the longer it took you to fall asleep, the worse your outcomes. And then additionally, the amount of time it took you to get to slow wave sleep also was related to worse memory outcomes. So I asked the question, are these sleep outcomes in part responsible for these poor memory outcomes we're getting the next day? So I ran a mediation analysis first with just regular sleep onset latency. I didn't see anything pair out there. When I looked at latency to slow wave sleep, I did find significant mediation, such that when I account for slow wave sleep in this model, the impact of stimulants on next day memory is no longer significant. And then this led me to the question, well, can I bring sleep back and then also bring back that post-sleep memory? So first, can we recover sleep with a hypnotic? The answer is no. We can't, generally speaking, for most of those outcomes. We were able to recover just slow wave sleep. So total sleep time, REM sleep, all those other outcomes were still gone. But we could recover slow wave sleep. And what we found here is that if we compare our stimulant condition, dex placebo, to our zolpidem conditions, we do see a recovery of slow wave sleep. So then we took that and said, well, does that also relate to next day memory performance? And so we looked first at test 2. What we found was just an overall effect of the hypnotic. So after sleep, if you have this hypnotic, you actually just perform better overall. Slightly driven in this placebo-zolpidem condition by negative memories. That has previously been found in the literature that zolpidem might actually increase negative memories. But we also see it here in the dextroamphetamine-zolpidem condition. However, that wasn't specific enough. I really wanted to see if we could recover this memory here. So then we look at the difference scores. What we do find is a little bit more of an interaction where the memory in the neutral condition for placebo-placebo is doing better than in the dextroamphetamine placebo condition and better than our placebo-zolpidem condition, and it's no different than our dextroamphetamine-zolpidem condition, suggesting that zolpidem was able to recover that memory slightly. And so with that, I'm going to just do a brief conclusion since I'm just about out of time. So the stimulant effects really do depend on cognitive domain. So for attention, we see these acute and delayed boosts to sustained attention, and stimulant sleep disruption doesn't seem to have as much of an impact on next day attentional processing. We were losing about an hour or so of sleep here in this particular design, and subjects had a lot of time to get some nighttime sleep. So if we can imagine if they had less time like we usually all do in the regular society, they may have had a little bit more of an impact here, but we didn't see it in this design. Working memory actually did not show any boost either in the acute or delayed conditions, but we did see some potential blocking of some training-related benefits that sleep actually encouraged. So that might be one of the kind of long-term costs that we were interested in. And then lastly, emotional memory had these really interesting impacts. So we saw this small boost to neutral information at encoding, making that information just as salient as some of our emotional memories. However, when we looked at our sleep disruption, that also came to a long-term cost to long-term memory processing. Just a really big takeaway from psychostimulants that they might have a slight short-term gain, small increases to attention that might even benefit memory. However, they do come at long-term costs to your nighttime sleep as well as your long-term memory. And losing sleep also has other types of health outcomes, so thinking about that in that domain is really important. And because I'm out of time, I'm just going to thank all the wonderful people that were involved with this project. This particular project was funded by the Office of Naval Research and tons of really great people across the UC system, Irvine, Riverside, that really helped support me in this work. And then my amazing advisors here at UC San Francisco, Dr. Eric Prather, Dr. Mendez, and Dr. Crystal, who have really been great in supporting me up to this point. And thank you all for your time and attention, and I'm happy to take questions. I did. So we asked them explicitly if they thought that they were, and people were actually quite good at it. So most of these analyses are controlling for that participant guess. But yeah, so participants, we asked them great, funny responses to that. People had really interesting feelings of self-empowerment after they were on the stimulant. One person said that they felt like they could go anywhere and ask any person out and they would get a yes. So really interesting responses. But yes, you did ask that question, and a lot of these analyses are controlling for that. Yes.
Studies. What about, I have two questions. One is, what about their sleepiness when they come in to start?
Yes.
It seems like if you're tired at the beginning, that's like, people use these drugs because they're like distracted by other stuff, they're having fun.
Yes.
And then what about boredom? Like,
Yeah.
if you really made them bored by the thing, would you see a bigger effect of the stimulant?
That's really interesting. So the first question is, we controlled for their sleep prior. That was one of our major questions is, do these stimulants help when you're sleep deprived? And if you're not sleep deprived, will they still have the same effect? So we controlled for everybody's sleep. Everybody was getting about 8 hours of sleep per night before they came into the lab, and we were controlling that with actigraphy and sleep diaries. So we wanted to make sure that they were really well rested before they came in.
Was there a relationship? You said you controlled for it, but was there?
No, so there was no variance there. Everyone was getting enough sleep. So that was one of our questions, but we didn't really tease that. So we could look at studies where people sleep deprive them and don't. The second question is actually really interesting, and Nora Volkow at NIDA has a really interesting theory about that when it comes to these psychostimulants. And what she suggests is that it's really about a salience effect, that if people are bored, these drugs are going to make things more interesting. And when they make things more interesting, that's really going to drive these different performance outcomes, which is kind of the story I was kind of using when it came to kind of the salience effect with the memory encoding.
Even in his article, like, party super interesting, 5 minutes after using cocaine.
Yeah, exactly. Exactly. Right. Exactly.